Hello, my name is Seth Gammon, and today I'm going to talk to you about small animal X-ray imaging, its various applications using the in vivo DXS Pro. As a brief outline of the webinar today, first I'm going to introduce you to the in vivo DXS Pro, then talk about some interesting X-ray imaging applications as they relate particularly to preclinical animal models, and then talk to you about a unique opportunity found with the in vivo DXS Pro, the ability to upgrade from X-ray to multiple modalities such as fluorescence, luminescence, and radioisotopic imaging. You see on the right here the in vivo DXS Pro. It's available from Carestream Molecular Imaging worldwide. It's a small animal and specimen radiography X-ray system. It's designed to be utilized with, it uses soft X-rays in order to optimally image small animals and specimens. The DXS Pro has a complete solution including acquisition and analysis software as well as the instrument and service provided by our service log. So here's a peek inside the DXS Pro. In this case, we're illustrating the instrument with a mouse. You see on the right here one of the key pieces of this technology, which is a patented, very thin, very uniform radiographic phosphor screen that in X-ray mode slides under the animal. The microfocus X-ray head turns on, illuminating the mouse with a color to visible light by the phosphor screen, where it then enters into the closed optical path, hits a 45-degree mirror, and gets passed to our true 16-bit digital camera. There, the data is quantified and can be imaged. The images can be exported, or you can quantify regions of interest in relevant units like X-ray density. The system also comes with multiple aluminum filters in order to allow you to optimize the bone to soft tissue contrast. As you get thicker aluminum filters, you see more and more of the bone relative to soft tissue structures. The ability to change and set these aluminum filters as well as the maximum X-ray energy are all controlled by the computer and can all be instantly recalled by the software force. The system also features a 10x optical continuous zoom. Thus, you can image a whole mouse, or really up to 5 mice, at our widest field of view of 20 centimeters by 20 centimeters, and then zoom in on key features of interest, such as the knee joint as illustrated here. There's also an optional geometric magnification stage that allows you to combine both optical zoom and geometric magnification. For those of you not familiar with geometric magnification, this is when you move the specimen closer to the X-ray source. Essentially, this is like what happens if you put your hand in front of a movie screen and move it closer to the projector. The size of your hand gets larger as you move it closer to the source. You do pay some small price, though, which is that the outline of your hand gets fuzzier. So you get a contact with the geometric magnification to get a quote final resolution, but that's simply not the case. Here's an example of a housefly taken with geometric magnification, and you can see quite nicely all of the many of the internal structures of this fly, as well as fine features like the wings. The DXS Pro is ideal for both rapid, high-throughput X-ray imaging. You can take exposures as short as 10 seconds that yield excellent image quality, such as you see here on the right. Or you can take longer exposures at higher f-stops that lead to superior image quality and indeed can lead to our optimal better than 25 line pairs per millimeter contact resolution. Reading the 10-second binning 2 image, as I showed before on the right, and the longer acquisition 2 minute with binning 1 image here on the left. The instrument is also designed for low dose, so you don't have to worry about the X-ray interfering with some biological process that you're trying to study or engaging some DNA repair pathway to any significant extent. For example, here we looked at a 30-second exposure, and the maximum absorbed dose in rats is 0.46. So at even shorter acquisition times, this goes even lower. Really, there's no negligible, the effect is negligible of the radiation on the system on your animal. So now we'll move on to introducing some of the applications one can achieve with the DXS Pro. One of the most obvious, and the best place to say, imaging system. We're all familiar with doctors looking at bone breaks or changes in bone development in standard film and digital X-ray. With a system like the DXS Pro, you can actually apply those very same techniques to small animals. And there are a couple key pieces of technology that enable that. Again, the thin uniform phosphor screen, the micro-focused X-ray head, and soft X-ray energies are all required in order to see these fine bones, for example, the metatarsals in a mouse pup. And actually, you can even track the growth and calcification process if you look at the 3-week-old mouse pup versus the 1-day-old mouse pup. If one were utilizing conventional, say, human X-ray systems, the energy of the X-rays would be way too high. You'd simply blow through these low calcification bones, and you would see terrible contrast if you could see these bones. Here's an example of looking at mouse phenotyping via X-ray. On the right, we have a knockout mouse that they wanted to find out what it was doing. You can see the significant developmental defect in the femur plate and growth compared to the one on the right. No quantification of this data is even required. Other developmental models include the zebrafish, which is a very challenging system to X-ray, again, because its bones are not very highly calcified. There's a very small sample. In this case, using the geometric magnification sticks, one can readily image both the soft tissue structures as well as the bone structures of a zebrafish at multiple developmental stages at the same time. Another interesting application is to look at how can we better improve bone repair. So one could imagine designing new and interesting metal plates and implants in order to better set and heal bones. And in fact, there was a group during a demonstration at Case Western Reserve that was looking at just those applications. You can see quite readily the implant, and you can see the bone break. So for example, if in a traditional methodology, one may not know that your surgical implant was positioned correctly in the animal until the end of the study. Here with an X-ray system, one can readily confirm that you've put it in the appropriate place and that it has stayed there after you've sewn the mouse back up and it started running around being a mouse. One can also, again, zoom in and see things like the details of the surgical implant as well as the detail you can get out of the system. You're looking at the grooves on basically a watch screw that was used to screw these plates onto the bone. Another model would be to look at bone regrowth without the aid of implants, perhaps looking at how stem cells might enable more effective regrowth, or just to look at the time course of bone regrowth at different points of the body with and without different drug treatments. So this group had drilled a 1 millimeter hole in the tibia of a mouse at different time points, and one can readily both visualize and then quantify changes in X-ray density over time as this wound site is healed. There are other structures that you can image and track changes besides bone. These include the heart. If you look at a mouse heart, Certainly the mouse heart beats far too fast for us to capture a static image, so what you're really looking at is a time-averaged image of the heart size. But one can still trace this outline with our region of interest tool and get out sizes in pixels or in more relevant units like millimeter squared or centimeter squared. The lungs are also visible via X-ray because they're mostly filled with air. So they are far less X-ray dense than the surrounding rib cage and tissue. This also enables you to look at diseases that affect the lungs, particularly diseases that add fluid to the lungs where you see an increase in X-ray density from control versus experimental animals. This is easy to visualize. It's slightly more difficult to quantify as regions of interest have to be drawn or should be drawn between the rib cage structure. Otherwise, you'll shrink your dynamic range and add noise. An X-ray system like this is also ideal for looking at changes in tooth development or in dental research. Here's the image of a mouse head using our geometric magnification and optical zoom combination. You can make out the details of the individual teeth as well as the rest of the bone structure of the head quite nicely. I've talked a lot about mice because they're one of the primary preclinical animal models. However, you can utilize the X-ray to image and quantify changes in phenotype in a wide variety of specimens, including plant seeds, insects, or fish. One of the other nice tools that are available for quantifying changes in phenotype is our bone density software. There's another to highlight this briefly. As published by Visit et al., the X-ray system can take a 2D X-ray image and for long bones make the reasonable assumption that the long bone is made up of two concentric cylinders at varying tissue depths. Then, because the X-ray system is unique in that it's a fully energy calibrated X-ray system, the software can go in and extract the parameters such as tissue depth, the radius of the bone, the radius of the marrow, and the densities of both the bone and the marrow, all from just a simple planar 2D X-ray. And you don't have to go through complicated three-dimensional drawings and renderings as you might have to do in a CT image. You just draw a simple box around the long bone, click measure, and you get a. Certainly one advantage of a CT system would be the ability to look at bone density in non-cylindrical tissues such as the spinal cord or the skull. But if one is interested in looking at bones like the femur, the tibia, or the metatarsals and tracking those changes in bone density, this will certainly do what you need and will do it a lot faster. Mostly we've been imaging and talking about imaging hard structures in the mouse, with the exception of the lungs. With contrast enhancement, one can also image softer tissue structures found in your small animal models. Here's an example of someone looking at gold contrast agent that was injected into the lower GI tract. One can also utilize barium sulfate, mix that with peanut butter, give it to the mice, and nicely highlight the stomach or lower GI. If one is interested in looking at the kidneys or the heart, you can utilize our ports that are found, our light type ports that are found in the side of our X-ray cabinet. And to do that, you feed a catheter into the instrument, you catheterize the mouse, close the door, take a pre-image of your mouse in X-ray mode, inject the contrast agent, have the software immediately begin capturing more images, and then subtract the subsequent images from the pre-image in order to highlight where the contrast agent has gone. This can then be overlaid onto the original image and color-coded in order to really highlight the kidneys and the heart. One could also make movies of the time course of this, how this contrast agent moves from the heart to the kidneys to the bladder as well. The vascular structure of animals can also be highlighted through the use of various veins in vivo, and then this image is an ex vivo image of the mouse after it's been sacrificed, but then one can nicely make out the vascularization of these regions, not only the major structures, but also the branching points and many of the fine points of the vascular system. This group also recently published a very similar study in Archives of Physiology and Biochemistry to look at vascularization in heart failure models. This group induced heart failure through aortic banding and then looked out at 3 weeks and 8 weeks to look at how the vascularization changed. At 3 weeks, there was an initial increase in vascularization, but by 8 weeks, the vascularization had significantly decreased. One can also see from this air failure. In order to image these, the mice were injected alive with contrast agents and then imaged both in vivo and ex vivo. These images that you're looking at are ex vivo images of the heart. They were then able to quantify these changes in vascularization by selecting random regions on the heart and quantifying changes in X-ray contrast. They made an interesting discovery. They hypothesized that by inhibiting various anti-angiogenic pathways, they would be able to recover vascularization in heart failure models. Indeed, one can look quite readily at the image here of the MMP9 knockout mouse and see that they were able to completely recover the vascularization of the heart during heart failure. Good old classic molecular biology was also utilized to confirm that MMP9 in the normal heart failure state, or the normal mice, not the knockout mice, was both present and highly active by 8 weeks. If you look at MMP2, it's a different story. There's pro-MMP2 at 3 weeks, but really you don't see the active MMP2 coming up very much. The DXF Pro offers you the unique opportunity to upgrade your instrument as your research needs change. So, let's say you're acquiring high-quality X-ray images, you're able to track interesting phenotypes in bone growth or bone destruction or bone development, but now you need to know more. In that previous example, what if I want an animal at the same time I'm tracking my vascularization? You can upgrade the DXF Pro to fluorescent, luminescent, and radioisotopic imaging and take advantage of the many in vivo molecular probes that are out there that report on those biochemical pathways in vivo. A lot of people really just want to use the X-ray as a co-registration modality for tracking fluorophores. So here's a group that wanted to develop an intranasal drug delivery system, so they added a near-IR fluorescent label onto the peptide. They had the mouse inhale it. You can see the nose is quite bright, as you'd expect. And then it goes into the head. And with co-registration, you can readily see that this is tracking to the olfactory bulbs. This type of study, I think, is a very exciting one, where they're looking at an arthritis model, which you can see the change in phenotomal animal. And then they injected, in their case, a proprietary fluorophore that gets activated at inflammatory sites, particularly at arthritis. You can then directly co-register this change in the molecular pathway with the change in phenotype of the bone degradation, both in space and in time. Here's another group that was interested in looking at the ability of the X-ray system to provide them better anatomical landmarks for their molecular imaging pathways. They wanted to look at inflammation induced at the lungs. As we talked about earlier, you can quite readily visualize the location of the lungs in an X-ray because they're much less dense than the surrounding rib cage and soft tissue. So they injected these mice intranasally with LPS, which induces acute inflammation. They then IV injected a activatable fluorophore that distributed throughout the mouth and then hopefully lit up at the site of inflammation. And in fact, what you can see from this image is there are 3 focal sites of inflammation occurring on this mouse from the fluorescence image. But you really don't know where that's coming from. If you overlay it on a white light reflectance image or a photograph, you start to get a better idea that it's in the upper torso. But really only when you co-register it with an X-ray image do you see that the one hot spot of activation is clearly coming from the lungs, whereas the rest of these activations are coming from the liver, which is found underneath the rib cage. So only with the X-ray co-registration do you get the nice complete picture of your imaging information. They then went on and did the right experiment, which is to confirm ex-vivo that in fact they were seeing the same thing, just as you saw in the in vivo image. Other groups were conducting surgery on bone structures such as the spine. In this case, this group injected clinically approved ICG into a surgically treated rat. The rats were then serially imaged, and ICG retention was then imaged. The idea was that if there was local inflammation around the surgical site, then the ICG would be retained. And in fact, they were able to do just that. They were able to see their X-ray images at the surgical site, and then they were able to visualize that their fluorescence they observed from the fluorescence image was coming, in fact, primarily from the surgical site, whereas without the X-ray co-registration, this would certainly be in doubt. So in conclusion, the DXS Pro offers you unmatched image quality with better than 20. It also offers a fully calibrated X-ray instrument, the only one on the market, that not only allows you to study and quantify changes in X-ray density over time or versus control versus experimental animals, but also to quantify changes in true bone density, all without having to get a CT system. You can image your choice of specimen sizes using our 10x optical zoom, or use it to zoom in on features of interest without having to resort to geometric magnification. The instrument, also unique amongst X-ray instruments, grows with your research. If you need to move from phenotypic X-ray imaging to molecular imaging, we're there to support you by doing field upgrades of the DXS Pro to fluorescence, luminescence, or radioisotopic imaging. If you need more information, please visit us at mi.carestreamhealth.com, or for questions, you can email me directly. Thank you very much for your attention again, and I'll be glad to take any questions.